welcome everyone. My name is Elina Zuniga, and I'm here with two of the co-organizers of the Global IMMUNOTALK, Dr. Enrique Veiga Fernandez and Dr. Carla Rodley. And we are all super excited to welcome and look forward to the talk of our fantastic speaker today, Dr. Miguel Soares, who introduced by Enrique. Before I give the mic to Enrique, I would like to remind you that our next global immune speaker is Dr. Fiona Powdy. And so with that, Enrique, I'll pass on to you.
So thank you so much, Elena. It's my real pleasure and delight to introduce our global IMMUNOTALK speakers of today. Miguel Soares, a good colleague and friend from the Lisbon area. So Miguel actually he graduated in biology by the University of Leuven in Belgium. It doesn't matter really the date, but then he went ahead and obtained his master and PhD also from the University of Leuven in Belgium in 1995. And after that period, he then moved to the US, most specifically to Boston, where he really worked with Fritz Bach, specifically at the Harvard Medical School. And there they really made a pioneer discovery that I think really shaped the career and the way of looking to science of Miguel. They've really identified tissue damage control mechanisms that actually were implicated in how transplanted organs could prevent their own rejection. And there, one of the findings were the fact that they realized that transplanted cells, tissues, or organs could induce the expression of the so-called stress-responsive enzymes, specifically a molecule called Heme 1 or HO1. And actually, these led then to a certain number of discoveries and research lines that Miguel has been pursuing over the years. So currently, Miguel is based in Lisbon at the Institute Gulbenkian of Ciência, of Science, or IGC. He joined IGC in 2004 as principal investigator, when he also became invited professor at the Lisbon Medical School in Portugal. So when he started his own independent research, one of the central questions, and I think we're going to hear a lot about it today, was really to interrogate first how disease tolerance is triggered and how can it be harnessed in the treatment of both old and emerging infectious diseases. Miguel really has been leading this research topic to the highest level. And it comes to no surprise that he extended this context in very important diseases such as malaria, or in the case of sepsis, for example. More recently, he also has been involved or exploring the role of the microbiota and how microbiota can regulate or enhance disease tolerant to infection. And actually this principle or his findings actually were pioneered for the development of anti-malaria vaccines, and this in joint effort with Bill and Melinda Gates Foundation. So I will not go through the many prizes and accolades he received, but you will see by yourself that his work is at the highest level, so it comes to no surprise that his work has been published in very important papers such as Cell, Science, Nature Medicine, Science Translational Medicine, and all the time around this very important context of disease tolerance. So Miguel has double nationality, Belgian, Portuguese, let's put it this way. He's currently based in Portugal. And I have to say on a personal note that for me it's a tremendous privilege to have Miguel as a close by, a nearby colleague that really works just next door. And it's a tremendous pleasure. So Miguel today will talk about the Iron Age of disease tolerant and infection. Miguel, thank you so much for joining us and for preparing the talk for the Global IMMUNOTALKs.
Enrique, thank you very much. And Carla and Elena, Enrique, you made me blush. I don't blush very often, but Thank you for your presentation. Let me try to share.
Before we move ahead, I mean, we wanna get to know you a little bit better.
Okay,
so we
sorry.
have a couple of questions. A simple question, not only to know you a bit better, but also I think that's also very, very important to inspire, you know, the young generation around the world of immunologists. So we would have a question that is, in your view, at the moment, what do you think is the most exciting future direction in the inflammation field that really should inspire young immunologists?
You prepare me for that, but the answer is difficult. I think the most inspiring question, it's going to be very individual. It's the question that every different individuals will feel it's the most important for them. And that's what they should follow. And they should follow that question because they cannot stop from doing it. That's what works in their heart, and that's what they... So I think answering that question is very biased because we answer what we follow with our hearts. So I am particularly interested on how we make sense of all of this, you know, at an organismal level, without abandoning the molecular biochemistry rules and tools that are required. I think we can now do molecular physiology, and I'm particularly interested on the physiology of inflammation and of infection. We know the rules of physiology for over a century at steady state, but steady state doesn't exist. Steady state is perpetually interrupted by rounds of infection, sometimes bigger, like right now, sometimes smaller, and we don't understand how inflammation and the rest of the immune system is integrated with the remaining of the organism to limit the fitness costs of our perpetual interaction with microbes. So it's a very, very broad question, and then in our case, we try to address it in the specific context where we think we can contribute. But I would go back to the answer, follow your heart. That's the important question.
Wonderful. What a wise advice to the young generation. So without further ado, Miguel, if you wouldn't mind to share your screen, and the stage is yours. We look forward to hear your talk and your science.
Can you just give me a... Is it working?
It is working.
Thank you. So once again, thank you very much for...
It is showing...
It's in presenter mode,
Okay.
Miguel. So if you...
Is it working? Is it working now?
There you go.
That's perfect.
Wonderful. Thank you very much.
So first of all, thank you very much for putting this... Let me just do one thing because there's one mistake here. Just give me 1 second. I'm sorry. I'm going to stop sharing. I think I need to share.
Not a problem. Take your time.
Can you tell me now if I'm sharing correctly?
That is perfect.
Okay, very good. Okay, so I want to thank you not only for inviting me, it's a real honor to be here, but for putting this these seminars together in this difficult period. I would, as a joke, I would say this is one of the positive things of COVID. It's that this type of activity. So you are really to be congratulated. So I work at the Gulbenkian Institute, which is a science institute. I call it science because it's very transdisciplinary. This is all of us in a good day, and it's in the outskirts of Lisbon. And I call the seminar the Iron Age of hosts of disease tolerance to infection. And I have a disclaimer, which is the Iron Age quote here is from my dear friend Marcelo Boz, and I keep using it. And every time I do it, I feel guilty about it. So what are we talking about? We're talking about defense strategies against infection. We want to understand how, as a whole organism, we limit the fitness costs imposed by microbes that are pathogenic. And they're pathogenic because they impose high fitness costs. And among immunologists, we don't need to dwell a lot into it because we say, okay, that's the role of the immune system. We have cells, innate and adaptive, of the adaptive of the innate and adaptive immune system, as well as soluble molecules that can sense and react upon the presence of pathogens to ultimately destroy them, if nothing else. And that's very efficient. That's how vaccines work and so on. Over the years, and I would say over the 150 years, we first realized that plants don't really deal with pathogens like that. They limit the fitness costs of infection, but they don't aim specifically at killing the pathogen. They just endure it. They just live with it in a way. And the original way this defense strategy was coined was disease tolerance. There's people that call it different names. So this is a defense mechanism which is as important as resistance to infection, but it carries one specific feature, which is it does not exert a direct negative impact on the pathogen. As you will see, like everything else, things will get a little bit more complicated. So how can we try to explain this? So here I plotted on y-axis health and fitness of a host. Arbitrary, this is 10, so very healthy, and 0 is dead, let's call it like that. And again, arbitrary, this is the pathogen burden of that host in a log scale from 10 to 10 to the 9. So as we get infected, as a pathogen manages to cross a barrier, an epithelial barrier typically, it starts growing, it starts getting nutrients that actually allow it to grow and multiply exponentially. And until a certain point, it does not really carry a negative impact on the host. It keeps growing, but the health of the host does not really change. After a tipping point, you see this curve coming down here. You get a mild disease, and then there's another weird point, which is here, that the host is starting to get a severe disease, but the curve now inflects to the left. This was actually, I didn't invent this. This was put forward the first time by David Schneider, and I think I advertised more for it than David, but I think they're really, really useful, these trajectories. So the curve inflects because there's resistance mechanisms of classical components of the immune system. They are now reducing pathogen burden, as you can see here. But this comes at a cost. Instead of getting healthier and healthier, this particular individual is getting more and more sick. Now, to recover and to regain, let's call it his initial status, you need the recovery phase here. So if you look at an individual, for example, that has 10 to the 4th pathogen burden, if it's here, you need to treat it perhaps by killing the microbe. But if it's here on the disease trajectory, killing the microbe is not the problem anymore, because actually it came all the way down several logs up. It's already clearing it, but there's something else missing here. And these typically are diseases that kill a lot of patients, traditionally severe forms of malaria caused by Plasmodium, sepsis caused by polymicrobial infections, and more recently, COVID-19 caused by SARS-CoV-2. They all carry these type of features. So everything that brings this system from the right side to the left side, that is, that reduces pathogen load, we refer to it as resistance mechanisms. Again, this is the job of immune cells that sense, target, and kill pathogens. I put it on purpose in the lower part here, because even though you're clearing, you're not much better off afterwards. But what you need to do is to go up this hill, and that's disease tolerance. So let's continue to try to see what is this elusive disease tolerance. Now here, what I try to do is to put very classically the way we see host pathogen interactions. So there's different classes of pathogens, they're sensed, they trigger immune activation. I try to put components of adaptive and innate immunity here, and they trigger effector functions. They exert a negative impact on the pathogen. So that's resistance. Like everything else in life, it comes with a price, the presence of pathogens and the expression of specific virulence factors, which, by the way, very often are there to capture nutrients, they're pathogenic. And also many of the inflammatory molecules and the effector molecules used by the immune system also carry a trade-off in which they can cause injury or immunopathology. Now, the sum of all these things is that you cause different levels of stress and damage to tissues. And this is usually something that we don't really worry about. But if you think about, for example, in a lung infection, these tissues would be the lung epithelium. So this only makes sense to clear the pathogen if you can keep breathing. Because if your breath goes away, you're dead. You can kill the pathogen, but you're dead. So you must sense this stress and damage and react upon it. And we refer to that as tissue damage control mechanisms. So those are the mechanisms that act in a cell autonomous or non-cell autonomous manner to make that the vital functions of those tissues and those organs persist. And for some of those, this is absolutely critical. For example, the lung, we are very used to hear that now with COVID, but the heart, the kidneys, and so on. Now, we put forward in this article here that this is under the control of what we call in a very convoluted way, transcriptional stress and damage response network. And I'll come to that in a second. And ultimately, this is what drives disease tolerance. So disease tolerance is very downstream of the normal playground that we usually deal with when we're thinking of host-pathogen interactions. Now, what is this elusive transcriptional stress and damage response network? This is a very simplistic view. So we plotted here, I think, 16 transcription factors, and each one of them responds to one specific form of stress. For example, some here respond to oxidative stress, hypoxia, unfolded proteins, and so on. Though if you look at what genes they can transcribe, genes that are only regulated by this transcription factor, some of these transcription factors have a fairly interesting private life, like P53 here, but some have a very dull life, like this one here only transcribes 6 genes. Now, but stress-responsive genes, they have different DNA binding sites for these transcription factors, and here is a group here that has at least 2 of these transcription factors regulating their expression. Now, if we go a little bit more inside, this is 3, 4, 5, 6, 7, or more. So there's a hierarchical structure here in that there's a group, a restricted group of genes that reacts to almost all types of forms of stress. So these transcription factors here, they congregate to transcribe specific things for specific situations, but some things are always transcribed. And I'm going to be talking about some of those. So the first question perhaps to address this is this just theoretical, or can we actually put some data and some facts into it? So the question is the establishment of disease tolerance to infection dependent upon individual master regulators, transcriptional master regulators of this network? And we think that's the case. So for example, if you take out this transcription factor called NRF2 that responds primarily to oxidative stress. So we can analyze this in different contexts. We like to analyze it in infectious diseases that carry a big burden to humanity, let's call it like that. And sepsis is definitely one of them. It's a multifactorial disease, so you get infection here, and you have also organ dysfunction. And sepsis is at the intersection of these 2 events. Now, by definition, it's a life-threatening organ dysfunction caused by dysregulation host response to infection. That's the definition that we all use now. And a very provocative paper actually suggests that this is nothing else than one of the major causes of human mortality and morbidity among any other cause. So this might be much more of a silent killer than we already suspected. And because it carries this organ dysfunction, it really calls upon to ask whether, this tissue damage control mechanism is regulated by the transcriptional stress and damage response network will be operational. So the experiments are fairly straightforward. So you take one mouse and you take that mouse at steady state. They're maintained in specific pathogen-free conditions, controlled temperature, controlled water, and you knock out one of these transcription factors, NRF2. Here we did it globally, so if you sample like 6 mouse, and then we challenge it. So this is a fairly rude approach to cause sepsis. You do a ligation of the cecum and you puncture it. And what you do is that to allow all millions of bacteria that are in the cecum now to come to the peritoneal cavity in an uncontrolled way, linked to also damage to the epithelium and cause a fulminant type of infection. And we use a lot of tricks here. So we treat with antibiotics, and this actually this one is not published, but we've published work with this. And you see actually that when the mice have the transcription factor, they're fairly resilient, tolerant to this, and you'll see why I call it tolerant. That ability to survive the infection is completely dependent on the activation of the transcription factor, because when we knock it out, you see that about 80% of them succumb to the infection. Now, this is associated with profound dysregulation of systemic culprits of metabolism, and glycemia is one of them. And we see here that without the transcription factor, you drop more on glucose. But the reason why we refer to this as disease tolerance, it's because when we compare the pathogen burden in different organs, including the peritoneal cavity, the blood, liver, lung, kidney, and we went for aerobes and anaerobic bacteria, we don't see a difference between surviving or dying. That means that this transcription factor, we don't really know where it's operating at this point, is actually providing a stress response that makes this host much less likely to die, but that can be dissociated from its ability to kill the pathogen. So this is by definition a pathway involved in disease tolerance. Another setting where these defense mechanisms are operational in a very strong way is malaria. Malaria is a disease that affects millions of people. It's an ancestral disease. We don't manage to have a vaccine against it. So we're doomed to understand why you get sick and why you die, and to try to find treatments. Unfortunately, it affects these younger children, like in this picture here, and there's a little mosquito there, and then it's a zoonotic disease, like SARS-CoV-2. These are the worst ones because the pathogens are not adapted to the new host. So they're like an elephant in a glass shop. Every time they move, they break everything. And this one here, Anopheles mosquito, he's after our blood. But in the process of doing that, he salivates, and when he salivates, he's himself infected with sporozoites from Plasmodium falciparum. That drops them in the skin, they go into the liver, they spend some time in the liver, no signs of disease, but then they go and invade red blood cells. And as they invade and they proliferate in red blood cells, that's where all the signs of the disease appears. So intuitively, one would say, but this has not always been the case, there's something really big price to pay to destroy red blood cells in the context of a systemic infection like this, and I'll be talking about that. So we did some very tangential clinical work in collaboration with people, but most of our work is really with mice. So we modeled the disease. This disease is actually a very good disease to model because you can change the genetic background of your mice, and you can change the genetic background of Plasmodium. And if you're interested on disease, you can just inject them with the infected red blood cells and then wait to see what happens. And what happens in these mice, in this background, when they get infected with this Plasmodium strain, is that they get a lot of the hallmarks of the human disease. And I'll point out acute kidney injury and liver damage because that's some of the outcomes that you will see coming in a second. So what would happen if we delete again the same transcription factor? So first of all, let me tell you what happens. What are the hallmarks of this experimental system? You infect them with the red blood cells, as I said. And if you look at parasitemia, which is the percentage of infected red blood cells and the red blood cell count, it's really incredible. So this is the percentage of infected red blood cells. These guys, they go up to 50% of the circulating red cells get infected, much more in humans, okay? But this is an experimental model. This is proof of concept, it's not. So if you look at the total number of red blood cells, they almost lose all their cells. This is a log scale. So they can withstand tremendous anemia. So it's a very exaggerated model, but it's a model that allows us really to look at extreme cases and try to understand some of the hallmarks of the clinical condition that occurs upon infection with Plasmodium falciparum. So again, the same trick, we delete specifically this transcription factor that responds to oxidative stress. We look at survival of this mice. And what we found, this work has been published, is that the wild-type mice that activate the transcription factor actually survive the infection, but that's completely dependent on that pathway, because if you delete it now, you die. And if you look here, we're looking at parasitemia, but you can look at different ways. There's absolutely no change in the percentage of circulating red blood cells, on the number of circulating blood cells. You can also go to different organs and so on, and we don't see changes. So this is again telling us that the activation of this transcriptional program is critical for this animal to survive Plasmodium infection in the conditions that I told you. So why would he die? That's a more difficult question to answer. So we can look at serological markers of organ dysfunction, like we do clinically, LDH. You see here that after infection, you don't really get an increase in the normal conditions, but if you don't have the transcription factor now, you increase. You have this for this liver damage marker, increases tremendously, so it's protecting the liver from damage. Creatinine kinase, so it's protecting the muscle, most likely, and urea is making the kidneys function. So we found one transcription factor that seems to fit the role that we ascribed to them, and we said, oh, it's going to be great. We're going to just, identify all 16 or 18 of these transcription factors and see exactly what they do. And the things might be far more complex than that. So let's call them transcriptional master regulators. And this has been, in a way, frustrating because we didn't get the data that we expected, but that's how science works. So what about this CHOC factor 1? So it sits here, okay, transcribes a multitude of these genes. It's classical damage response. I call it damage response because it reacts to unfolding of proteins, which is already damaging the structure. It's absolutely critical to maintain homeostasis, so one would expect to have a lot of damage or a lot of unfolding of proteins, and therefore this to be absolutely critical. So we used more technological ways to delete in an inducible manner this transcription factor in adult animals. We infect them with the same Plasmodium strain, and we're going to look at deletion of the gene survival in parasitemia. So the deletion is great. This is we look at peripheral blood monocyte, circulating cells, leukocytes, I'm sorry. But then we don't see any change in survival. There might be more subtle changes, but nothing apparent. And again, nothing apparent on, on the pathogen load. So here we got shocked. We said, well, perhaps we didn't look at the right one. So let's look at one that everybody's looking at for years now. It's really important. It reacts to hypoxia. And we have anemia, so we should have tons of hypoxia. So this transcription factor in the stress network should really be important. So we went through the ropes, as we said. We got the mice. We delete them again globally. We put them through the same system. Deletion is beautiful. It has been described by many other people. But again, no change whatsoever in survival. And no change in pathogen load. So we thought about testing the 16, but we were afraid of having 16 disappointments. And so perhaps it's more complex. Perhaps what this is telling us, and this is my interpretation, is that the transcriptional master regulators, they're just there to be able to activate transcriptional programs to different forms of stress. But probably what counts is what they converge upon to. And in some situations, this is going to be very specific, but some other situations, these are going to be core effector genes that are important for this. So another way of dealing with this network is to say, what are the core genes that are there? And it turns out that one of the core genes here is the gene that we had been working for quite some time, called heme 1. And what I'm going to do now is to go back a little bit in time and explain to you what is this gene and why do we think it is important and how to interpret this data. So, so it's a heme catabolizing enzyme, and why would the heme catabolizing enzyme be at the core of a network that responds to stress. It looks very obvious, but hopefully I'm right on what I'm going to tell you. The answer to this is really on the substrate. And the substrate is Heme. So Heme doesn't rotate like this. I just put it like that so you won't forget it. But what's important there, it's the yellow atom in the middle. And the yellow atom in the middle is an atom of iron. And I'll show you in a second, most of the iron that mammals have is inside Heme. I can even be more precise in a second. And this is a really smart evolutionary conserved way of intercalating iron into the 3D structures of proteins that allows amino acids to control the reactivity of the iron, and it allows the proteins to acquire redox capacity and exchange electrons via the control exchange that this divalent metal allows for. Now, a couple of little dirty secrets about iron bioavailability. I'm going to stress it again. Almost all the iron we have is in Heme, right? And we always think about iron like this. So catabolic pathways for Heme, like the ones I'm going to be describing, they turn the system this way, and biosynthetic pathways of Heme put it this way. So where's this Heme? 70% is in hemoglobin in red blood cells. By the way, that's where Plasmodium likes to go. Myoglobin takes about 5 to 10% of the Heme, and cytochromes and other proteins take the rest. And each one has its corresponding cell. Red blood cells for muscle, for myoglobin will be muscle, and any cell produces Heme because it has a variety of hemoproteins. So a few years back, and based really on the work of my dear friend Marcelo Boza, who has been studying this for a while, we wrote an opinion article together, and I'll stress again that this is really based on his work. And the message is that we decided to write a red alert. Labile is an alarmin. So it's a prototypical alarmin. It's a molecule that should be inside cells, and once it gets released, it alerts the immune system for something that is profoundly wrong. So Heme can be released, for example, from hemoglobin, much more difficult from myoglobin, because here in hemoglobin is non-covalently bound. So if you oxidize the protein, it gets released. The work of Marcelo and his colleagues has identified pattern recognition receptors in different innate immune cells that are actually sensing labile and providing an appropriate response to it. And even endothelial cells by the groups have been shown to do that. So one of the things that cells do immediately when they see Heme, it's to activate, they induce the expression of the stress-responsive hemoxygenase 1 enzyme that opens the protoporphyrin ring of Heme here and extracts the iron away. But it also produces this gasotransmitter carbon monoxide. It requires electrons that it's getting from here, and then it produces this really cool and interesting molecule called biliverdin that we'll not be talking about today. So this is really traveling in time. When I was working with the late Fritz Bach, for a whole set of reasons, we started transplanting mouse hearts into rats. And we were doing that, and we were treating the rats with a specific immunosuppressive protocol that allowed these hearts to survive long-term. But what we found was that the survival of the transplanted organ was completely dependent on its ability to induce the expression of hemoxygenase 1. This was very, very positive. And what we found really was that when the heart does not express HO1, we see a lot of cardiac myocytes and endothelial cells that undergo programmed cell death, probably by apoptosis. And we identified that hemoxygenase 1 was actually a cytoprotective gene. Now, when I moved to the Gulbenkian Institute, I started working in a variety of other systems, including malaria that I'll be talking today. And we identified with the group of Maria Mota that actually this gene was critical to protect against one of the most prevalent forms, lethal forms of malaria, called severe malaria. So mice survive malaria, but if you take some strains of mice, do not develop experimental severe malaria, but they need to express HO1. With Angelo Soares, student at the time in the laboratory, he identified actually that hemoxygenase 1 was critical to tame down the severity of autoimmune neuroinflammation, the model of VAE. You can see actually that once you delete the expression of the gene, it becomes much more severe and lethal. we identified with Rasmus Larsen, same type of things for sepsis. The group of Claudius inclusion in Germany identified that the expression of immunogenase 1 is absolutely critical, actually, to have a viable pregnancy, I'm sorry. You can see here that if you don't have HO1, you actually, in this model, you go from abortions of about 10% to almost 100%. And even for more chronic lesions, such as atherosclerosis, this is absolutely critical. Now, why would you do that? There's many ways to do this, but one of them is by carbon monoxide. So if we go back to our transplant model, and if you take again these ARTs, but now transplant them and use pharmacological inhibitor of immunogenase 1, and if you look at the percent of graft survival, you see that these grafts don't survive because you require enzymatic activity actually to sustain the survival of the graft. Now, if you require the enzymatic activity, you probably require one of its end products. And what we found here is that if you put carbon monoxide via inhalation at very low dosage, you now completely recover. So you go from here, it's kind of magic. All these ARDs now survive long term. And you reverse the programmed cell death that you were seeing in the myocardium that becomes now completely viable just by the presence of carbon monoxide. This led to the identification of this gasotransmitter being a very strong cytoprotective molecule. Now I'm going to do exactly the same type of scheme. It's carbon monoxide that is actually preventing cerebral malaria. It's carbon monoxide that is reducing autoimmune neuroinflammation. This is not our work for sepsis. This is a work of Mark Perella and colleagues at Harvard Medical School using CO-releasing molecules to tame down the severity of sepsis. Again, in the Claudius inclusion, to reverse spontaneous abortion, CO actually does the job very, very well. And back in the days, we identified that for restenosis, this was extremely potent in stopping these chronic inflammatory lesions that go from year to year. So why would carbon monoxide do that? One of the reasons why it really does it, and it's very simple, is that it binds to iron in the Heme groups of the hemoproteins that are releasing the labile. So again, the problem is the labile. And once it binds to the iron, it doesn't allow it to oxidize, and it doesn't allow the Heme to work in a variety of pro-inflammatory and cytotoxic ways. So basically, if you put carbon monoxide here, this whole thing disappears. So perhaps the next question could be, why is labile so bad? And why does it contribute to these bloodstream infections, to the pathogenesis of these bloodstream infections? So I'm just going to go very briefly to some old work in sepsis. We found out that once you infect a mouse and do again the sepsis model of cecal ligation and puncture, first of all, we're looking at the wild types here and we analyze the expression of HO1. You see a lot in the peritoneal cavity where the infection starts. You also see a lot in the kidney. Now, if you delete this gene globally, you now see that animals that otherwise survive, they succumb to the infection. They do so even in the absence of adaptive immunity. So what we're trying to do is to say this is not dependent on adaptive immunity. And the pathogen load, again, this was done in a more not so sophisticated way in the time, but we couldn't see any differences between them. So I told you, I made a claim for carbon monoxide. It's again a red blood cell releasing hemoglobin, oxidizing, releasing its labile in, and being captured by immunogenase 1. But if the problem of Heme, as you will see, is iron, then you're releasing iron out of it. And this iron can actually be very pro-oxidant unless it gets trapped by a protein called ferritin. So I'm going to call this the peroxidase component of the ferritin complex, FTH, and I'm going to ask whether it contributes to establish disease tolerance to infection. And this is what I wanted to say, is that this is a post-transcriptional element associated with that transcriptional network. So the system is again the same. We do a loss of function approach. This is not completely total. It's an DMX group promoter. We did cecal ligation and puncture, and we see actually that susceptibility to sepsis increases tremendously. This was the work of Sebastian Weiss when he was in our laboratory. We see again no differences whatsoever in pathogen load, aerobes, anaerobes in different organs. These mice are treated with antibiotics, so they're really reliant on something else than killing the pathogen to survive, which is very reminiscent of the real problem that sepsis imposes. And I'm making a very long story short, we were desperate. We were desperate because we couldn't find anything that would justify why this iron regulatory molecule, which was already world famous before we started even playing with it, could be so important. And truly by desperation and by intelligence, Sebastian decided to look at glucose. And what he found was that there is a drop of glucose 24 hours after the induction of the septic shock. Actually, there's an initial rise that comes down, but this graph only shows this. And then the animals that survived then kind of recovered, they linger, but they recovered. But these ones, we noticed, Sebastian noticed, they were really going down. So we asked what could be happening, and I couldn't make sense of this. So with Sebastian, we kind of designed an experiment that would go straight to the end and say, look, if regulation of iron metabolism is really regulating glucose, and if you see hypoglycemia, just give them glucose. It doesn't make any sense, but let's give it a try. And what Sebastian found was very astonishing to us still today, which is you knock out this iron regulatory gene, but just by providing glucose, which is what they were missing, now they all survive back. And again, this comes as no surprise now, there's no change in pathogen load. Now, why would regulation of iron metabolism do this? And why would regulation of iron metabolism by ferritin do this? So we did target metabolomics, and what we found was that once you don't have ferritin in the liver starts promoting glycolysis. So it takes the glucose to feed the Krebs cycle. But once you have ferritin, it switches back to produce glycolysis, which is one of the things that the liver must do. So we have a switch between this and this that is regulated by ferritin. So we did RNA microarray at the time, and we found a very weak signal, but significant for glucose 6-phosphatase C1, and that begged the question at the time, what is glucose 6-phosphatase C1? And we went to Stryer and other lab books, and it said the obvious, which is the last enzyme in the gluconeogenesis pathway that is absolutely required to take phosphate from glucose 6-phosphate and allow glucose to be secreted. So that to us begged the question whether hepatic glucose production was at all required to establish disease tolerance. So we collaborated with Gilles Mithieu in France, who had been studying this enzyme for many years, and he was very generous and just collaborated with us. And we had a mouse in which it cannot produce glucose from the liver because we deleted glucose 6-phosphatase. We did another model of sepsis, and what we found is that these mice really develop hypoglycemia. You cannot survive with this, and obviously they're succumbing to sepsis. So this tells us something that we didn't know, which is to survive sepsis, you need really to sustain in some way glucose production in the liver. Again, no change in pathogen load. So let's go back to Heme, because we kind of lost track of it, and ask what is the relationship with Labile and gluconeogenesis. So it turns out that Labile represses glucose 6-phosphatase C1 transcription. This is an experiment done by Xian Wu in the laboratory by QRT-PCR. You take primary mouse hepatocytes, you expose them to Labile, and you can get these beautiful dose responses of Heme repressing glucose 6-phosphatase. Now, this is an experiment done by Anahita Carlos some time back, where she does a luciferase assay to analyze more specifically transcription, with or without ferritin, and is exposing the cells here in epithelial cell line to Heme alone or Heme and TNF. I'm just going to show you Heme and TNF. And what she found initially is that if you put ferritin, you get more transcription of glucose 6-phosphatase. If you give Heme and TNF, you repress it, but the repression is diminished. It's not abolished, but it's diminished by ferritin. So one of the roles of ferritin might be to stop the deleterious capacity of Heme to stop gluconeogenesis. Now, even more astonishing, at least to us, is that what would happen if you inject Heme? You're not doing sepsis anymore. You're just injecting Heme to an animal that cannot produce glucose from the liver. Now let's look at their glycemia. They drop glycemia to levels that are non-compatible with life whatsoever. And here we don't need to measure pathogen load because there's not even an infection. We'll see this again in a second. So we came up with this model that we think stands, which is polymicrobial infection triggers metabolic response, which I'm not referring to, called anorexic infection. You stop eating, you stop absorbing glucose from diet. And the work of Ruslan Metzintov's lab with Andrew Wang, did some, again, marvelous work. And they actually had shown that you need to drop glucose once you're infected with bacteria to survive. So this was very puzzling. And what we think is happening is that you need to drop glucose, but you need to control it. And you cannot go below a certain threshold level. And that's actually the role of ferritin to stop this effect of IM and now allow it, the system not to go all the way here when we don't have ferritin. And by having ferritin, then you get the system to impose survival. So is hepatic glucose production required to counter the pathogenic effect of the pathological outcome or the pathogenesis of other bloodstream infections? The obvious question is obviously malaria. So for that, we had to do a strange, difficult cellular-based reporter assay because it's very difficult to detect labile IM. So we put a reporter into cells, we bring IM, and we see a peroxidase activity that these cells have because they are transfected with an phosphorylated peroxidase. And I'm not going to go through details. These are actually the control cells. And once you give them IM, they go all the way back here, and you can see actually the proteins that get IM incorporated and they acquire peroxidase activity. So it's very tedious, but this is IM that it's not bound to proteins and can actually become bioavailable into cells. So does it accumulate really during Plasmodium? And it does. There's actually, let's just call it tons of it there. This is IM that it's unlocked from hemoglobin and can actually be accessible to different cell compartments. This is an experiment we did very recently, and I decided to share it with you because I find it really exciting and puzzling. And so what we're asking here is, what is the difference between being infected with Plasmodium and IM? And everybody would say, oh, it's a world of difference, one would expect. Plasmodium is much more complex than injecting a molecule. So the readout for this experiment is RNA-seq from liver, bulk RNA-seq. And I'm just going to go fast through the slide for the sake of time. But here we're comparing the induction of genes by IM versus the induction of genes by Plasmodium infection or their repression by IM. Oops, sorry. So everything that sits here is shared. What I'm trying to tell you is that most of the transcriptional regulation, I don't know if I can say most, but a very significant fingerprint of the transcriptional regulation associated in the liver with Plasmodium infection can be mimicked just by injecting IM. And there's a component that represses genes that can also be mimicked. And in a snapshot, so if you look at all the regulated genes regulated by Plasmodium or by IM, about 40% are shared. And it's a big number here. So if you look at the upregulated, it's 30%, and the repressed is about 40%. So, I'm going to exaggerate a little bit, but 40% of the gene transcription profile of associated with malaria is most likely strongly influenced or directly mediated by IM. Right. So one of the genes that is here, I forgot to tell you, is actually glucose 6-phosphatase. So we did the obvious, which is to ask whether hepatic glucose production is required to support survival to the blood stage of Plasmodium infection. And as you can see, it's absolutely required. When you can't produce glucose, you actually succumb to the infection. I'm not showing you the details for the sake of time, but you develop this tremendous hypoglycemia, and it does not change pathogen load. And I want to finish the talk by complicating things. And I had the privilege to see Janella Ayres last week. And I think this really talks into some of the concepts that she's putting forward. And I think we're finding the same things, which is really exciting, at least to me. So before that, I want to show you, I showed these disease trajectories that David Schneider put forward. I want to put them into practice here. So these are the control mice that can produce glucose. This is the number of infected red blood cells that they have. This is the amount of glucose that they have. This is normal glycemia for them. as pathogen load increases, the black lines, each one is one animal, they drop a little bit at maximum pathogen load, and they go immediately back to the normal. But that's if you can produce glucose in the liver. If not, you fall into what David called this death zone here. And that's exactly what we're seeing. You go here, you develop hypoglycemia, and you die. And then you can start looking at other parameters. This seems to be absolutely critical, and we now know why. I don't have time to discuss it, to maintain core body temperature. It's not critical, actually, to maintain weight, or it's not critical to actually avoid anemia. So it's very specific. So I want to finish by sharing with you also some unpublished data that, again, we find very exciting, which is, I'm putting this word here, behavior of pathogens. And you will see in a second what I mean by that. So what I mean by that is this idea that Janelle showed you last week of this collective behavior of host pathogen interactions. And depending on the behavior of both sides, an interaction that is pathogenic can become actually non-pathogenic because the pathogen changes its behavior. And we think this is happening here. So if you bear with me for the last 5 minutes. So this is a mouse that is infected with Chabaudi, as I showed you before. He will die because he cannot produce glucose from the liver. But here we measured red blood cell count, hemoglobin, and hematocrit, so hematology parameters. And we noticed something strange, which is when you don't have glucose, you actually have more red blood cells compared to these animals that can produce normal levels of glucose, but they do a more profound anemia. So we checked it in many different ways, and indeed, at the peak of infection, when you don't have glucose, you have more erythrocytes, you have more hemoglobin, total hemoglobin in blood, and you have more hematocrit. So you really have less anemia. This is an hemolytic disease where Plasmodium is actually lysing red blood cells. So we start wondering what is happening here. So we took a careful look at the maximal percentage of infected red blood cells that an animal carries through all the disease trajectory. And it turns out that actually, when you don't have glucose, you do less percentage of infected red blood cells, but you do more red blood cells. And therefore, when you multiply this by this, you don't see a change in pathogen load. So there's probably changes in behavior, in collective behavior, that are not actually allowed to be perceived if you just look at pathogen load. Now, we've been looking very actively into this now, and I call it, I don't know if I can call it like this. Let's look at pathogen behavior. So Plasmodium is interesting because you can really look into this because it's inside red blood cells. It's this ugly, some people like it, parasitologists will find it beautiful, but this is kind of organism inside a red blood cell here. When you have glucose, you're not normal glycemic, but you're not profoundly hypoglycemic. Look at the animals that cannot produce glucose from the liver. They do these dots, and these are pycnotic parasites. This is a very big change in the physiology of the parasite. So we quantified this, and it's actually Wilson and Susanna that have been doing this, and there's a tremendous difference between these two conditions, producing glucose from the liver or not, in terms of pycnosis. And when you don't produce glucose, actually these parasites become pycnotic or start dying, which is very consistent with the literature saying that these parasites, they absolutely require actually glucose to multiply and survive. But the other astonishing thing is that they switch the program and they don't like the host where they are, so they want to get away. So they start activating a program that says, let's become a gametocyte. And for Plasmodium, that's the only way out of this host. So we see this big difference here again, from almost no gametocytes in animals that survive and produce glucose, but Plasmodium senses the lack of glucose and probably other things. We think temperature is very important to actually transform itself into a very big number of gametocytes. Now, another feature of this collective behavior changes is that you favor transmission in detriment, often, not always, of virulence. So you don't, if you want to transmit, you cannot kill your host. So we tested that in many ways. I'm going to show you one. So the way we tested this is that we infect a mouse that produces glucose in the liver or not. Okay, this guy is doomed to die. But before they die, when they're hypoglycemic, we take the parasites and we now infect another mouse that cannot produce glucose. So the environment, the pressure is always there on the Plasmodium. And this was actually done by Wilson and Gilles. And what they found, I'm sorry I didn't put Gilles' name here. And what they find is that when you take actually parasites from a mouse that was hypoglycemic, the virulence of that parasite is much reduced. It kills 50% less about as compared to the normal parasites, but it's not because of changes of pathogen load. So it's this collective behavior changing, favoring transmission in detriment of virulence. And this was previously found for bacteria, for an enteric bacteria. And it's pretty incredible that this can be translated to a protozoan parasite. So it looks as if we collectively find cardinal features of host pathogen interactions. So I'll just finish it up here by telling you that disease tolerance is an evolutionary conserved. And evolutionary conserved is a tricky name. What I mean to say is that it's really important because it's conserved. We cannot get rid of it to defend ourselves against infection. And regulating of host iron metabolism is absolutely essential to control glycemia in the context of bloodstream infections. And another cardinal feature that we're seeing is this interaction between glucose metabolism, either because you produce more or less glucose or because you consume less or more glucose, and probably there's a control in both sides. And I think we have data for that as well. I wouldn't be showing you any of this if there wasn't a fantastic lab to thank. We are the inflammation laboratory at the IGC. Now I cannot say most of the work that I've shown you because I've really covered years and years of work, but I would like to show Sebastian Weiss and Rita Carlos, who is here, who did all this sepsis work. Susanna Ramos, that I referred to many times, she's here. And I mean, everybody contributed in a way or another, and I'm just thankful to them. I think I will stop here. I have 3 minutes too much, and I thank you very much for your attention.
Thank you, Miguel. That was beautiful, your talk. It's really fascinating how whole body metabolism integrates with disease tolerance and the outcome of infection. Yeah, thank you so much.
Thank you.
Insights. Okay, I will share the screen again now, just to guide everyone to ask questions for Miguel via Twitter. And so if you have questions, search for the account Global IMMUNOTALKS and find the tweet that says ask questions for Dr. Miguel Soares here. And Miguel will reply to that tweet with your question, and Miguel will use his personal account, Miguel Soares, to answer your questions. And so remember, you can ask questions right after, or if you watch the talk via YouTube, you can ask questions after that. Okay, thank you so much again, Miguel. Thank you so much, Enrique, for co-hosting this session. And yeah, I hope to see everyone again. We'll see next week for our Global IMMUNOTALK by Dr. Fiona Powry.
Thank you very, very much. This was
Thank you. Bye-bye.
real pleasure. Thank you. Bye-bye.
Thank you.
Appreciate it. Bye.